Sorry for the delayed start. It's the classic interoperability thing. You know, you turn up and actually it doesn't interoperable. But there we go. So I'm Hugh Glover, architect, developer, software builder. Done various things over the years. Probably about 30 years worth of that. But you'll be wondering why there's a dog on there. Well, in the UK, there is a set of children's stories written by Beatrix Potter. And this is John Joyner. And John Joyner goes around with his basket of tools and mends things. What all my family know me for doing is fiddling around with the boat and mending the boat. So at home, I'm always known as Dog. So you'll hear the cry around the house, Dog, Dog, come and mend this. So that's me. What I want to talk about the next 30 minutes is clinical research and the processes that go into it. It's sort of parallel universe compared to clinical care. And some of the things that go on seem like they're the same things, and some of them are the same things, but other things are just sort of actually just viewed from a different perspective. So what I want to talk about is the business processes that we go through. And I'm looking at this as much as anything from an opportunities point of view, because this is a market which people are not getting involved in. It's a market where people are just starting to say, FHIR. Somebody mentioned that to me the other day. And I think there is an opportunity here for this sort of group to be thinking about the opportunities for building small apps, building bits and pieces, and starting to get into that, because I think there is going to be a big increase in this market over the next few years. And towards the end of the presentation, I'll talk about why I think that's going to expand. I also want to talk about the various groups that get involved in clinical research as a whole, particularly from within HL7, but also how some of the other external groups get pulled into that. So you've got a bit of a sense of what the landscape is. So let's start off talking about a clinical trial and how an actual clinical trial works. So your first step with a clinical trial actually is even before this. You have to get approval from the regulators. So you'll have to get an approval from the EMA if you're doing something in Europe. If you were just doing something in the UK and nowhere else, you'd be looking at the local regulator. If you're doing something in the US, you'd be getting approval from the FDA. If you were going to Japan, you'd then be looking at the JPMA and so on. So you've got to get your approvals first, but once you've got your approvals, they'll say, yep, we approve you doing a trial over 15 sites in at least 3 countries. So you set about finding your sites. And you know, your sites will build up over a period. This is not a whiz-bang, we've done it today. This is something that you set out to do, and you have to go out and you have to find the sites. And the other question is, who's actually going out and doing the finding the sites? If you've not been involved in this industry, you tend to think that, well, it's pharma that goes out and does it, you know. It's Pfizer and Bayer and all the rest of them. That's what I thought when I started getting involved in it. In reality, they may have some impact on it, but most of the time they contract out almost all of this work to the clinical research organizations. So it's not the drug company that wants the trial doing that's trying to sort this out. It's the other companies that are going to manage it for them. And they have a set of standard sites, standard investigators they will go to. And what typically happens is that they'll come up with a series, and if you counted the stars, there's actually 16 on there, not 15, because they've got one more than they asked for in the first place. Oh, that's fine. So now they go through and they start saying, well, do all these sites actually meet the requirement? Because people will come along and say, yeah, I can do your trial. And so you then have to go back and check the resources. And a classic example is that you're going to need a freezer to store things at minus 29 degrees. So you have to go and validate that the freezer will actually do that, but the freezer doesn't actually do that. So that site actually can't take part in the trial. So you started off with 15, you started off with 16, now you've lost 2. And then you start going through and you start doing your recruitment. And so the numbers that are filling in are meant to be the number of subjects that you've actually got on each of these trial sites. But as that goes through, you then go and look at the subjects, and you start finding that, well, it doesn't say it in there, but some of our subjects must not have a history of asthma. And you go and you start to talk to the subjects and go through the initial phases, and you find, well, actually, 3 of the subjects that came in, although they didn't admit to it, it's very obvious that actually they have got history of asthma, so they come back off the list. You also find a few sites, there's one here, they've only managed to recruit 2 subjects. That's so few, it's not actually worth having them in there. So you fiddle around with this, and you end up now, well, we've got a trial where we've got, I think that's 13 sites, and you've got 180 subjects. So it's not what you started off with. That's typically the case. And yeah, this is okay, we can go ahead with the trial. Nothing in that has talked about clinical data. But all of that is processes where you're flowing information backwards and forwards. And I've said, oh yes, it's a CRO that's going to be running this for the drug company. It's not necessarily the same CRO running it in France as is running it in the UK, as is running it in Germany. So you might have 3 different systems just running your trials in the first place. So there's data transfer got to happen within that. Now you've got to start supplying drugs to your trial. Different trials work in different ways. Some trials, the people will actually be at home. So they'll get a supply of drugs will come at home. They'll get a week's worth delivered at a time. And they don't deliver more than a week's worth because this is expensive stuff and you don't want to lose track of it. So there are then systems which will allow people to either go to a web interface or to a telephone interface and say, I need some more drugs, please. I've used up all the lot I've got. Within that process, there's keeping track of the usage of those drugs. So you don't want somebody ringing you up every 2 days and saying, I need another 5, please. You've got to be able to come back and say, well, no, actually, it's too soon for you to be wanting more. You've got to flag that back up, that this is a subject who is not following the protocol, the things are not going as they should do. Again, you've got data being transferred backwards and forwards in there. Then you go off to the stuff that we tend to think of as what happens in a clinical trial. You start gathering data. You're taking classic blood pressure, temperature, and everything else. Then you go off and you do some analysis of the data, and then you submit the data to the regulator. Well, now we've got into a whole different ballpark, because now we've accumulated all this data. Oh, and by the way, it's been collected by 3 different systems, and now we've got to bring it all together. And those 3 sets of data are amalgamated, then they have to be sent to the regulator for the regulator to approve it. So again, a lot of transfer of data and moving stuff around. So what I've got, this is the sort of background slide that's going to build up, and I'm going to put other stuff on top of it afterwards. So it's not meant as something as a perfect representation. But there are different classes of software that go on. So there's one set of, press this one, this work? No. So this line here, there's a set of software, clinical trial management systems, a standard piece that gets used. It does things like getting investigators, getting sites, keeping track of the investigators who have actually done a good job of what they're doing, particularly keeping track of the investigators that utterly fail in what they're supposed to do. You know, the investigator says, I can get you 20 subjects. They never do. They manage to get 1 subject, manage to get 2 subjects. They would go on a blacklist. This set of software keeps track of that set of people. Again, we've got individuals, we're passing data around about them, we're keeping registries of them. This lot here, this is the logistics side of it. where you store your products, the people ringing up saying, I need more drugs, keeping track of what's happened to them after you've sent them out. When the trial ends, have you actually got as many left over as you would expect to have? If not, where have they gone? Because you've got to keep track of all these things. Data capture, well, this is the classic sort of stuff that we tend to think of. And then adverse events, big deal. Adverse events are in clinical trials. It's what we're looking for more than anything else, something that's gone wrong. So you've got separate systems for dealing with that. All of that has got to go up into some central data store. The items that we're dealing with, so we've got subjects, we've got investigators, sites, countries. We've got this interesting distinction between what you expect to have happen and what actually happens for an individual. So I've said that as part of the protocol, the expectation is that on the for your first thing that happens, well, the second thing, so you've got the first thing that happens, so that's maybe going just to be assessed in the first place and get your first dose of the drug. Your second dose of the drug happens 10 days later, and the next dose of the drug is another 10 days after that. In reality, you're trying to do that for a real patient. The first dose of the drug is on a Monday. The next dose of the drug is going to happen in the middle of the week, the following week, so that's fine. If you shift your days along, and my brain can't do it on the fly, but if you move that along 10 days on from that first dose is on a Sunday, and the patient's not going to turn up on a Sunday, so it's actually 11 days, and you have to work that through. And there will be other things going on. So each individual subject will end up with their own version of the plan of what's actually going on. So having done all the management, kept track of all that data, which is using this data source up here, you're then going to have to pull that in, and it will have to go off into the separate data set for doing the analysis. That's typically done by an arcane set of software that the pharma industry likes to use for these things. We're not in a position where we're going to change the way they do that. That's the way they like to do it. And then it has to be submitted, and it has to go through a set of protocols again. Oh, look, here's clinical care. Now, this is all going on at the same time. You know, particularly if you've got somebody who's on a trial who's in hospital, you've got all the top lot of stuff going on for that patient at the same time as you've got all the EHR stuff, which is what most people in this session have been talking about all the way through. And that data is going into a different repository. So that's our framework. That's the world I'm talking about, and that's where it fits. So where do all the resources actually drop in on this? Where do the FHIR resources go? There are two main resources that people think of at the moment when you talk about clinical research within FHIR, and that's research study and research subject. It's pretty obvious what they deal with. I'm not going to go into the fine detail of that. There is a sense, I think, that, ooh, these are very, very basic resources. They're very draft, they're very unstable. They're not something that we really want to risk making a lot of use of at the moment. The reason that attitude is around at present is, I'm afraid, because they weren't terribly well looked after by their own in committee when they were first put together. That has very much changed now. But those resources are sitting at maturity level 0 at the moment. In reality, they're probably more like a level 2. Though research study 1 is being used by Accenture to keep track of all the clinical trials that are being registered in the US. That's all going into an RDF database, making use of that, and they've been using it for that for the last 2 years. That's not an immature resource that doesn't meet any actual need. It's one that the committee failed to register what was going on and push it far enough up the list. That's not to say that these are not changing. We're looking at research subject at the moment and making some changes and some extensions to that. But the expectation is that over the next 6 to 9 months, we'll be pushing through a fairly detailed review of those, so that when we get to the R5, these will be stable resources, at least at level 3. Down here at the bottom is IDMP. Now, you won't find a resource called IDMP, but how many people were at Rick Smithy's presentation? Right, so the majority of you have no idea what I'm talking about. IDMP is identification of medicinal products. There was a project established by the major regulators, it must have started about 10 years ago, to get a standardized method of identification of all the drugs as they went through the trial process. This is what IDMP is about. It exists as 5 different ISO standards, and there is a whole set of attributes of the drugs that are required that are definitional, which are over and above the things that you would normally use in clinical care. In order to manage that submission process and the issuing of the IDMP identifiers, there needed to be a set of FHIR resources created. So these have produced a new set now, which have got their own separate block on the bottom right of the list. If you look at all the set of resources, there's about 7 of them there. So medicinal product definition, and I'm sorry, I can't remember what all of them are, but they're all going through and defining different aspects of the way a product fits. Those all get owned within that section of the product. Adverse event, so that's a standard thing. It's been used, 2 different sorts of adverse event. Adverse events, as they're seen within the clinical care setting, tend to be a different focus and a different emphasis, and certainly a different management process than adverse events as they're seen within clinical research. And then we've got the consent resource, because people have obviously got to agree to get involved in a trial. So there's a lot of different bits and pieces going on in that, and one of the challenges, particularly if you're trying to get things to move a bit quicker, is to coordinate the way those resources get used and how they get put together. So I want quickly just to sort of thread these together. So research study is your core bit. That's what everything hangs off. Your research subject is part of your research study, and research subject is related to a patient, but is not actually a patient. We'll come back to that in a minute. Research subject consents to something. Research studies happen at sites. And this is a bit of an oddity. I'll come back to that one in a minute as well. Research studies have sponsors, and they have principal investigators. So it may be obvious, but a sponsor is an organization, a principal investigator is a practitioner. A site is kind of sort of a location. And then we've got the protocol. Now, the protocol is the be-all and end-all of defining what happens in a clinical trial. It doesn't just say what the sequence of events are. It says how many patients you have to have in it. It says what all the treatments are. It says what the drugs have got to be. It says what the conditions have got to be before a patient is allowed onto a trial. So it's a big set of things. And that is the legal document that governs what can go on in a trial. So when you sit down and you try and say, oh, well, which FHIR resource does that actually fit to? It's quite difficult to pin anything singly down and say, oh yeah, that's the protocol. But research study has got this point that hangs off it, that's pointing to protocol. So protocol in FHIR terms is not the same as protocol in the normal clinical research terms. We're taking a much narrower definition, and it hangs off plan definition. Then, of course, patients actually go through their process of care and will be subject to a care plan of some sort. So I talked a bit already about protocol and the oddities. You know, there is no real equivalent to the protocol as a whole in general clinical care. But I mentioned subject and patient are not the same thing. The subject is an anonymous entity as far as a clinical trial is concerned. We're not actually, generally speaking, interested in patients. Patients are not the focus of a clinical trial. The point behind a clinical trial is to work out whether a drug actually does what it's supposed to do or not. That's what the objective is. And we also have this thing about blinding and permissions. I think they're less common than they used to be. But one of the standard things within a clinical trial is that you don't want people feeling better because they think they've been given something, as opposed to better because they've actually been given something that really does the job. So we all know about the placebo effect. You need to not know whether a patient is receiving your study drug or, a sugar tablet. So keeping the anonymity of what's happening is really important in many trials. Not all of them, but in many trials. That's the blinding process. So it's not visible to anybody who's involved with the trial as to what part of the treatment process or part of the plan a given subject is actually receiving. And then there's a whole set of processes. If something goes wrong and you need to work out what part of the process they're in, there's a whole unblinding process. So that's something you don't have an equivalent to in clinical care. And people go, oh yeah, but it's just the same as permissions. It's what you can see and what you can't see. Well, yeah, sort of. But it's all the baggage that goes with it and the processes that go with it. And it is seriously important in the trials that are blinded trials, because if the blinding breaks down and people start working out what it is, the entire trial collapses. So however many $1 million have got spent on putting that trial together just go out the window, together with all the results that go with them. So it is a big deal. As part of that, you also need to be very careful that you're not giving away patient information to people that shouldn't be receiving it. If I'm running a clinical trial and I want to do analysis on, how blood pressure has changed on a set of 50 people as a consequence of them being on that drug, I don't need to know what those 50 people are. I don't need to know any of their personal details. I need to know their gender probably. I probably need to know their age, but I don't need any more than that. And keeping that set of data separate from the clinical trial people is, again, an important thing. And that's why we keep subject and patient separate. Because if you talk about the subject and you only give subject data out, you can constrain what that data is much more securely than if you're saying, oh, well, you can have that field, but you can't have that field. Let's talk about information architecture a little bit. Anybody know what that is? Apart from a mess. Not you, Andy. I know you know what it is. Okay, this is the bridge model, the biomedical research and whatever it is. You can see it's horrendous. It's vast numbers of UML classes that set out to describe the entirety of the clinical research domain. This has been around for maybe 10 years or so, and particularly within some sections of the US, particularly the cancer care, they make huge use of this for organizing all of the data that's being pulled together. There are systems out there that are aggregating 50 years worth of trial data according to this as a structure. And it's one of the things that we've got a benefit of within clinical research, because we've got this underlying information model that talks about how the data fits together and what the core concepts are, what the main pieces are. But the question then is, how does that actually relate to FHIR? So we've got it as an underpinning to our entire process, but can we actually make some sense of it and help us to understand how we make use of FHIR? So that takes us on to then starting to explore where these different FHIR resources fit together. How many people here have made use of ClinFHIR or ConMan? 2 or 3? Okay. This is a tool that you can get. It's part of the, this wonderful ecosystem of FHIR tools produced by David Hay. It allows you to drag resources onto a canvas and link them together, and it will only link together the ones that are allowed to be linked together. So we have used that as a means of finding out what the best relationships are and how we represent the data. Because you can populate these resources you pull onto the canvas with actual data. And you don't need to do any software or anything, just sit there and build it up. If you haven't played with it and you're trying to work out how to use resources, I recommend it as a way of doing it. We have a mindset, because of that information model I showed you before, that if you're looking at the way things work, we think in terms of there being a definition of it, a plan for what would happen, and then the result, what gets performed at the end. So defined, planned, and performed. And we also think of it from a protocol, which has within it a schedule of activities, which has within it a single visit, which has within it 3 components: defined, planned, and performed. So that's the mindset that we've been adopting. And then we've made use of the ClinFHIR tool to actually build this up. And this is a piece of work that's been done by the Fuse group, and thanks to Jeff Lowe for the next set of slides. I am not going to go through these in any detail at all. But the point is that these are all snaps from ClinFHIR, looking at how we thread the pieces together. And so we've worked our way through and we've threaded all these different bits until we get to something that starts to make sense as a way of putting it together. So those are in the deck. You can go and look at those as a separate exercise. This is the structure that we had before. So we've got our plan definition. Definition, remember, is sitting off here. The care plan is actually performed, is what's happened. So how now do those pieces thread together? We think that plan definition really breaks into a number of different pieces. I think what we're probably going to end up doing is doing some profiles to pick those out. So there will be a profile to cover the study drug and a profile to cover the schedule of activities, quite possibly a profile to describe the treatment arm as well. And then within the schedule of activities, each of the individual activities, take vital signs, would be one of the things that hangs off that. The care plan ties back across to the activity definitions, but the care plan has also got within it greater detail. So if you get down to something single, like you've got a blood result being taken, that will actually tie back across to your activity definition. So now we've got this linkage in place, you can start to say that, oh, this observation here ties back to this activity definition, which is part of this schedule of activities, which was set up by the protocol, which belonged to this research study. And in this particular case, it was this research subject that was subject to it. So we start now to thread these pieces of data together. So now we can look at this and say, well, hang on a minute. We've got things going on here which are being captured, and we've got things going on here that are being captured. We've potentially got the same thing being happened. So you've got a trial in which somebody needs a full blood count being done, and they're in hospital being treated. What's the betting that they're going to get a full blood count being done at some point? What happens at the moment is that the patient gets 2 blood draws, 2 separate sets of analysis are done, and you've just got duplication and wastage, and you made 2 holes in the patient rather than 1. There is no technical reason why we shouldn't be linking these 2 lots together and got the mechanics there that will actually pull that together. So projects that are going on at the moment, I'm not going to go through this in fine detail. We've got one chunk of projects that's happened that's looking at mapping between actual data that's stored as part of the trial and what goes in the submission process. There is other chunks of work going on that are looking at using wearables to get things directly from patients into your trial record. You've got other projects that are going on that are looking at actually managing the investigators so that you're not repeatedly doing manual transfer of data. And then something I've not talked about at all, on the manufacturing side of things, all those IDMP resources are getting used for product quality as well. So lots of bits happening in there. But nobody's building a terminology server specifically for clinical research. Nobody's constructed a sample population for clinical research. Nobody has built any clinical trial management systems based on FHIR out of that. Nobody has done anything on the logistics part of that. Nobody's done anything on the data capture side of it. Nobody's done anything on the scheduled activities. And we haven't yet got the white papers in place to cover all the other bits. So there's an awful lot of stuff still there that can be built up. Genomics is not my speciality, and I'm going to jump over that. So BR&R is a committee within HL7. I'm one of the co-chairs of it. We look after all those resources on that part of the process. And as I say, we're now functioning, I think, a lot better than we used to. The other thing that is happening and what's driving this forwards a lot is a meeting that started off in Atlanta and which is going to bring together this great string of organizations, HL7, Translate, and so on, into a new accelerator project that's going to be looking at making use of FHIR resources for clinical research as a whole. So I hope that, DevDays 2020 in Amsterdam, I'm actually going to be coming back with a whole set of things that we've actually achieved and where we've got to. So I think I've gone through what I intended to. And the man at the back's been waving at me now, so I invite any questions. Okay, thank you very much.